Remember that I should take it back.
Okay.
Can you see if it's working? Click it here. Yes. Is it? Oh, okay, perfect. All right. Okay. So now we are starting with the new topic, the chemical mediators. You all know what is chemical mediators. So these are the chemicals which actually helps in the inflammatory process, right? So these are the substances which helps in the inflammatory process. So we have basically two main groups of chemical mediators. One are the cell-derived mediators, and the other is a plasma-derived mediators. So the cell-derived mediators uh, uh, are produced inside the cell. Some are preformed, and the other they form later, right? So further, we also divide them into two: preformed chemical mediators as well as the chemical mediators which are processed later when they are triggered. The plasma-derived mediators usually it's produced in the liver, right? In the liver. So the active mediators are produced in response to various stimuli, like bacterial product, which it enters, it triggers the chemical mediator to be produced. Substance released from the necrotic tissue, for example, anything which injures that particular area, that also provoke the inflammatory process of mediators. And protein of the complement, kinase, and coagulative system, they also pushes those mediators to go on. So one mediator can also stimulate and release the other one. For example, tumor necrosis factor, when it is released, it acts on endothelial cell, and then they start secreting other mediators like interleukin 1 and many other chemokines. If you remember that lecture, my first one, the acute inflammation, there's just two, tumor necrosis factor and interleukin 1, they go up and they stimulate the endothelial cell, and also they pushes histamine and thrombin to activate. And what they do, the histamine and thrombin, they activate the p-selectin and e-selectin, and tumor necrosis factor and interleukin 1, they stimulate integrins, right? ICAM, VCAM, right? Both on the endothelium as well as the cells, right? Then chemokine also, because later on chemokine attaches with the protoglycan, and then it allow that the integrin, which is in a low affinity, to the high affinity. The doors are open then, and they stick tightly with the ICAM or VCAM, so that they can be pulled in outside the blood vessels. Now, mediator vary in their range of the cellular target. So many have different effect on different cells. And the important thing is, once they are activated and released from the cells, they have a short time. So either they die itself, or there are some enzymes which usually kills or stop that chemical mediators. You had to stop the inflammatory process, right? It should not be forever. So as it is started, then it will be stopped. Now, what's the function of this mediators? Can I put it up a little bit? Here. Right. It's not okay. Okay, so they are signals that initiate the process. These chemical mediators signal, they are coordinate the process. So one in the chemical mediator will assist and activate the other one, so they go in groups. And finally, signal that turn off the process. The process which has been started for inflammation, it should stop, right? So these are the chemical mediators which we will be discussing, and also discussing individually. So as I said, there are chemical mediators which are secreted by the cell, this one, right? And there are, in the cells, we have preformed mediators, as well as we have newly synthesized mediators. So in a preformed mediators, which are present as a granules, are present as a histamine, serotonin, and lysosomal enzyme. Although we have discussed these things already, but we will be discussing more. So histamine are been released from the mast cells, basophil, and the platelets. Serotonin, they are usually been secreted from the platelets. They are also present in enterochromaffin cells in a GI. The whole GI has this serotonin. So they activate the movement of the intestine. And then lysosomal enzyme like neutrophils and macrophages. You know that lysosomal enzymes are the enzyme which kills the bacteria, right? We have discussed two different pattern of killing: oxygen dependent and oxygen independent. So this is oxygen independent type of killing of the bacteria. Then we have newly synthesized one, prostaglandin, secreted by all leukocyte, platelets, and endothelial cells. Then we have leukotrienes like all leukocyte, platelet activating factors. So all leukocyte and endothelial cells, activated oxygen species, all leukocyte. These are all activated species like the oxygen superoxide, OH, and hydrogen peroxide oxidase. So these are all oxygen species. Nitric oxide, macrophages, cytokines like they are secreted by lymphocyte, macrophages, and endothelial cells. So there are other group which is plasma derived, which is usually been formed in the liver. They are factor 12, which is Hedgeman factor. They actually forms this kinin system, bradykinin, as well as coagulative system, fibrinolysis system. The other one is complement. So the important complements are C5A, C3A, C3B, C5A, C5B, and then we have membrane attack complex, which is C6 to C9. Okay. Now, basically, what you have to do is you have to remember the name of the chemical mediator and what is its function. For example, if I say serotonin, what's the function of serotonin? It causes vasoconstriction, right? Histamine, what does it do? Vasodilatation. So this is important thing. So this chart actually will show that which chemical mediator is doing where, what, and where it has been secreted, and what are the important cells. You have to memorize all these things, right? Because in exam, you will be given a name of one chemical mediator. So you have to remember what's the function of it and where it is secreted from. Okay, so this chart will, and again, I have distributed the chemicals mediator name and the action so that it will be memorized in your mind. So histamine, they are secreted from mast cells, basophil, platelets. Their function is vasodilatation and increase vascular permeability, and they also activate endothelium. So histamine, these are the functions you should know. Serotonin, they are present in platelets. Again, the function is vasoconstriction and increased permeability. Prostaglandin, mast cells and leukocyte, the function is vasodilation plus pain and fever. So pain and fever is prostaglandin, which causes this, especially prostaglandin E2, right? Leukotrienes, they are secreted by mast cells and leukocyte. They have increased vascular permeability, chemotaxis, leukocyte adhesion and activation, right? This is the same leukotriene, which is a part of, when we'll come there, from where it is coming from. It's a part of arachidonic acid metabolite, and it secretes this leukotrienes. Then we have platelet activating factor, which are secreted by leukocytes and mast cells. The function is vasodilation, increased vascular permeability, increased leukocyte adhesions, chemotaxis, degranulation, and oxygen burst. Reactive oxygen, leukocytes, especially neutrophils and macrophages. Again, killing of the bacteria and tissue damage. Why tissue damage? As we were discussing acute inflammation, while it kills the bacteria, it bursts off. So when it bursts off, what will it do? it causes necrosis of the local area. How it has been cleared? The macrophages comes there after the process is end, and the function of the macrophage is to clean. Scavenger cell. It is called scavenger cell. Macrophages are also called antigen presenting cell, right? They are also scavenger cell. Their function is to clean everything. When we will be completing the inflammatory process, there is one slide which gives you a full detail how the things are going on or how the things are finishing on now. Then we have nitric oxide, which has been secreted by endothelial cell macrophages. The function is vascular smooth muscle relaxation and killing of the bacteria. This is the same nitrous oxide which comes from arginine, and you need enzyme which is inducible nitrous oxide synthase, and it allow this nitrous oxide to come in. It joins with superoxide and they form peroxynitrate, which is also one of the very strong killer of the bacteria. This is all present in your last lecture. Then we have cytokine like tumor necrosis factor and interleukin 1. They are secreted by macrophages, endothelial cell, and mast cells. Again, the function is local endothelial activation. You know about it, how this TNF and interleukin 1, they activate the integrins, which is ICAM, VCAM, and they stick the leukocyte together, right? Expression of adhesion molecule, you know it about. Now, this is another cytokine, the chemical mediator which causes pain and fever. So that's why I have colored it. So you should be remembering that one is prostaglandin, other is cytokine which is causing fever and pain, and it also causes anorexia, hypotension, decreased vascular resistance, and shock. Then we have chemokines. They are secreted by leukocyte activated macrophages, and the function is chemotaxis and leukocyte activation. Okay. Now, protein plasma derived proteins. What are those? One is complement, which is really important, and their function in inflammation is tremendous. The important one is C5a, C3a, and C4a, C3b. C3b act as a opsonin. What is opsin or what is opsonization? Yes, when it attaches to the bacteria, it attracts the leukocyte to come and eat it, right? Phagocytosis, good. Plasma, it's secreted from the liver, and it causes leukocyte chemotaxis, as you said, and activation plus vasodilation. So they are also called C3a and C5a. They are also called anaphylatoxins. Why? Because when these attach with the mast cell, it degranulate mast cell and secrete histamine and causes vasodilation. So they can even cause anaphylactic shocks. Yes. You know, they are of low potency, but this is an important one, C3b. C3b. If you just open your Robbins book, they have highlighted only C3b. So I have not gone into detail. I will show you a chart when we will be discussing complement. A complex chart is there just to make you understand. Otherwise, we will shift to the chart which is given in your book. So I have taken only those important things which is important for you. If you go in detail, then there are many other things which I'm not going to come in detail, right? Okay. Then we have kinins. They are also from plasma, derived from liver, are produced in the liver, and they cause increased vascular permeability, smooth muscle contraction, vasodilation, and pain. So this is the third one, pain, which is causing by kinins, bradykinin. So not more. So if you go back, pain, and the third one. The first pain is by prostaglandin, the second by cytokines, and the third one is this kinase. Then finally, we have proteases activated during coagulation. So these are also produced in the liver, and they causes endothelial activation, leukocyte recruitment. Okay. Role. So that's why I said it will be repeated again and again so that you will be memorizing those all. So they have, what are those causing vascular dilation? These are histamine, prostaglandin, nitric oxide. Now you can make a chart and then you fill more. Whatever there, you make a full chart. These are the chemical mediators which are doing vasodilation. Increased vascular permeability, histamine, bradykinin, nitric acid, oxide, prostaglandins, and leukotrienes. Then which promote leukocyte adhesion, which is interleukin 1, alpha, TNF alpha, complement, prostaglandin, and leukotrienes. Which causes promote chemotaxis. They are prostaglandin and leukotrienes. Now we will go individual chemical mediator one by one, not in detail, but you know something about it. For example, histamine, they are secreted from mast cells, basophils, and platelets. So it's this is important, that which chemical mediator is secreting from which side. Okay. They are powerful vasodilator. They increase vascular permeability. They are vasoactive amines. IgE, when the IgE come and attach to the mast cells, it degranulate and cause secretion of this histamine. There are other many substances like histamine released by the mast cell in response to physical injury like trauma, cold, or heat. Binding of antibody to the mast cells, Fc fragment, which also degranulate and secretion of histamine. Complement fragment like anaphylatoxin like C3A and C5A, we have discussed early that when they attach and they degranulate, histamine releasing protein derived from leukocytes, neuropeptide like hepcidin P, and cytokine like interleukin 1 and 8. Serotonin, this is another one. It is also called 5-hydroxytryptamine, serotonin. They are present in platelet, are secreted from platelets and also enterochromaffin cells, as I have already mentioned. These serotonin are enterochromaffin cells. They are lined, the whole GI tract is lined by these enterochromaffin cells. So yes, hello. Yes, this serotonin are where it is distributed. Yes, I have just mentioned now. So you are busy in your mobile. Put it down and be attentive here. When I see somebody is looking here and there, I'll throw a question on it. Don't do it. So this serotonin, they are being secreted from platelet as well as enterochromaffin cells. These enterochromaffin cells, they are present in the whole GI, and usually it regulates the movement of the GI tract. Then we have, yes. Sorry? 5-hydroxytryptamine, which is also called 5-HT. Yeah, this is a name of the serotonin. I think this is only one chemical mediator which has a specific name. I think it's better if you remember this. And it will be actually be helping you in GI tract, because there are few diseases in which it causes many problems. Then it present as a preformed granules. It causes vasoconstriction as well as increased vascular permeability. Icosapent, yes.
How is it vasoconstriction and increase?
Yeah, this is really the question, because if you open the previous Robbins book, it is written that they do vasodilation, and suddenly in edition 9 and 10, they have changed the function from vasodilation to vasoconstriction. So we are also confused how it is doing this. But because this is there in a book, we cannot deny it. You can go and open 8th edition Robbins. This says the serotonin do vasodilation. And suddenly in edition 9 and 10, it says serotonin, they do vasoconstriction. So we also say it's doing vasoconstriction now. Okay. Icosanoids are arachidonic acid metabolites. They have it is present in a cell wall, phospholipid area, and they are prostaglandin, leukotrienes, and also thromboxane, leukotrienes, and lipoxins. This is a new one, and the function of the lipoxin is to close inflammatory process, right? Now, why it is called icosanoid? Because icosa means 20, Roman one, right? That's why they are called icosanoid. So if you see the green round, these are the carbon atom. They are 20 in number. That's why it's called icosanoid. So please remember, it's very important. Sometimes arachidonic acid metabolite is replaced by icosanoid. So if somebody say icosanoid, you should not be confused. You should know that icosanoid is a second name for arachidonic acid metabolite. So if you see this chart, it will say that although it is present in a cell wall, cell membrane phospholipid, but it has to be activated through phospholipase enzyme. And this phospholipase enzyme is activated by different cytokines, right? So when it is activated, what it will do, it has two different pathways. One is cyclooxygenase and the other one is lipoxygenase. The cyclooxygenase pathway, they usually secrete prostaglandins, mostly. So in a prostaglandin, the important one is prostacyclin, which is PGI2. This causes vasodilation, inhibit platelet aggregation. So it is just opposite to thromboxane A2, which is another prostaglandin, which causes vasoconstriction, promote platelet aggregate, right? So when you need a clot in the blood vessel, then thromboxane A2 stimulate and they act. And if you need to take out the clot, to abolish the clot, then prostacyclin will activate. It will inhibit platelet aggregation and also causes vasodilation. Clear? And then we have prostaglandin D2 and E2, which causes vasodilation, increased vascular permeability. Also, it causes, you can add that, causes fever and pain, especially prostaglandin E2. Clear? Okay. The other part is now this portion, which is cyclooxygenase. It has been seen that aspirin and endomethacin, they actually block this pathway. So whenever somebody has pain or fever, we used to give him aspirin or endomethacin so that it blocks that pathway and patient does not feel pain or the fever comes down, right? The other pathway is lipoxygenase. In lipoxygenase, we have leukotrienes A, which convert into leukotriene B. And this leukotriene B is a strong chemotactic factor. Then leukotriene C4, D4, E4, they are actually helping in vasoconstriction, bronchospasm, increased vascular permeability. Along with this, they have another one, which is a new product of this arachidonic acid metabolite, which is lipoxin A4 and B4. These are actually inhibit neutrophil adhesion and chemotaxis. So once the neutrophils are inhibited for chemotaxis or adhesion to the endothelial cell, what it will do, it will not allow this neutrophil to come down. So this lipoxin act as anti-inflammatory thing, right? So if you want to stop this inflammatory process, lipoxin A4 and B4 helps for that, right? Okay, then what is the action of these? They cause vasodilation, which is caused by prostaglandin I2, which is prostacyclin, prostaglandin E2 and D2. Then we have vasoconstriction caused by thromboxane A2 and leukotrienes C4, D4, and E4. Increased vascular permeability, leukotrienes C4, D4, E4, and prostaglandin E2 and D2. And finally, chemotaxis by leukotriene B4 and HETE. Okay. So if you go back, this is all written in this chart. You memorize this chart, and if you just continuously see this chart, everything will come in your mind. So in complement or this arachidonic acid metabolite, You just have to memorize all this thing by looking at the chart, and then you will remember. One important thing is the steroid, because it has two different paths. So if you give aspirin or endomethacin, it will only block cyclooxygenase, not lipoxygenase. So lipoxygenase pathway will be remain open. But if you have to completely close all this thing, then you give steroid. It basically is stuck here, and it does not allow this arachidonic acid metabolite to be activated. So both cyclooxygenase and lipoxygenase pathway will be blocked. Okay. Now, prostaglandin and thromboxane, including they causes pain, fever, and clotting. Leukotriene, they causes chemotaxis, vasoconstriction, and increased vascular permeability. Lipoxane, they cause inhibit chemotaxis, so they actually are anti-inflammatory, vasodilation, and counteract action of leukotrienes. Coming to platelet activating factor, another one, very important. They are also present in phospholipid-derived mediators on the cell wall. Many cells like platelets, leukocyte, mast cells, macrophages, and endothelial cells has this platelet activating factor. In an initial time when we were students, uh, means after passing this MBBS, because in our time there were only two chemical mediators, two, when we did MBBS. One is histamine, another was serotonin. That's all, no other one. Now, slowly and gradually, it comes out, and it was thought that platelet activating factor is only present in platelets. That's all. But now they say that no, there are many other cells that have this platelet activating factor. So they cause vasodilation, increased vascular permeability. They are 100 to 10,000 times more potent than histamine. Just imagine, when they are activated, they cause vasodilation, which is more potent than histamine. So they cause leukocyte adhesion plus chemotaxis. They activate platelet powerfully. So at the time of when you need a clot, you need to have this one. Okay. Oxygen species, you all know from where this come. Oxygen dependent killing of the bacteria. Yes. So they synthesize via NADPH oxidase, which is phagocyte oxidase. Just remember that slide. They derive from neutrophil and macrophages. So there are two important cells which generate this thing, activated by macrophage, immune complex, and cytokines. So when NADPH, in the presence of phagocyte oxidase enzyme, this NADPH, they convert into NADP, and then this H comes in, it causes oxygen to develop into superoxide, and then the whole cascade starts. So you have superoxide, you have hydrogen peroxidase, you have OH ion, right? So these are all free radicals there. The important function is to killing of the bacteria, and they are very, very destructive. So they do the destruction of the surrounding areas. Although after inflammatory process, the whole thing will be cleared off. Nitric oxide, they are present in macrophage, neutrophil, and endothelial cells. They are potent vasodilator. So they also act as anti-inflammatory. So there are two chemical mediators. One is lipoxygen A2 and B2, and number two is nitrous oxide. So if I ask, what are the chemical mediators which stop inflammatory process? So you had to give two names, right? Okay. Potent vasodilator, they are produced from the action of nitrous oxide synthase. Again, you remember that this nitrous oxide synthase is there, and it takes the nitrous oxide from arginine, and this arginine, when it with the presence of inducible nitrous oxide synthase, they comes in, join with superoxide, and form peroxynitrite, which is also one of the powerful killer of the bacteria. Good. So not only killing of the bacteria, they also inhibit inflammatory response. So this is a second chemical mediator which stop the inflammatory process. Everything is going on well, clear. Please kindly stop me anywhere in the middle of my lecture if you don't understand anything, right?
What is the first chemical mediator?
Lipoxin A2B2, which is there in a arachidonic acid metabolite, two pathways, cyclooxygenase, lipoxygenase. So in lipoxygenase, the third one is lipoxin A4 and A4 and B4. So these are the mediator which stops inflammatory process. Okay. If you want, I can go back to show you. Okay. The lecture will be with you, right? It's on the Moodle now, and whenever you want, you can see my lecture. Cytokine and chemokines. So cytokines are the protein produced by many cells, but usually lymphocyte, macrophages, and endothelial cells. Numerous roles in acute and chronic inflammation. Tumor necrosis factor alpha and interleukin 1, they are activated. They activate by the activated macrophages. If you remember my slide in the last lecture, this antigen presenting cell, which is macrophage, when they engulf that bacteria, just after this, it secretes tumor necrosis factor interleukin. So macrophages is a very important area from where it comes. So it causes local endothelial activation, expression of adhesion molecules. You all remember that it goes up and they activate ICAM and VCAM integrins. It causes fever and pain, so you know it. Fever and pain, anorexia, hypotension, decreased vascular resistance, shock. Now, chemokines are a small protein, smaller than the cytokines. They have a smaller protein, 8 to 10 kilodalton weight, which are attracted attracted for the neutrophils more than 40 times, right? Also lymphocyte, monocyte, and eosinophils. And they are classified into 4 main groups. So we will be discussing each group now according to the arrangement of cysteine molecule. So we have number 1 is CXC. So the amino acid is present in the middle of 2 cysteine molecules, CXC, separate between the 2 cysteine. This chemokine, which is IL-8, act primarily on neutrophils. So they attract neutrophil more. So if I ask what is the chemokine for neutrophil, you will say CXC, right? So you have, because I have highlighted this thing. It's important. If you just see it, you will remember. CXC for neutrophil. Okay. The other one is CC. There is no amino acid between. So two cysteine groups adjacent each other, including monocyte chemoattractant protein MCP1, which is called CCL2, which attracts monocyte. Then we have eotoxins, which is CCL11, which attract eosinophils, and macrophage inflammatory protein 1 alpha, which is MIP1 alpha, which is CCL3, mainly serve as chemoattractant for monocyte, eosinophil, basophil, and lymphocyte. So CC chemokine can attract almost all the cells. But in this one, the important one is eotoxin. So eotoxin chemokine only attract eosinophil. So suppose if I ask which chemokine attract eosinophil, so you should know that it is CC chemokine. Okay. Now coming back to the third one, which is only C, 116. So this C chemokine, they attract, they have lymphotactin, which is XCL1, and they usually attract lymphocyte. So we have a different one. The first one, CXC for neutrophils. The second one, eosinophil, and other one. The third one is especially for lymphocyte. The fourth one is CX3C. Means in this chemokines, we have 3 amino groups between 2 cysteine. So 2 cysteine and 3 amino group. This chemokine promote strong adhesion of monocyte. So we have almost each and every, and T cell. T cell means lymphocyte also. So we have different chemokine for different cells. So if I say specifically I cannot say that only CX3C for monocyte, because definitely CC is also promoting that one, right? So just remember that these are the chemokines which attract which cells. Okay. Now, the actions they are really important in both acute and chronic inflammation. So the tumor necrosis factor, as you know, secreted by macrophages, mast cells, and T lymphocyte, they stimulate expression of endothelial adhesion molecule and secretion of other cytokines, and they have systemic effects. So what are the systemic effects? We will be looking after forward, right? In the next slide. Interleukin 1, they are also secreted by macrophage endothelial cells and some epithelial cells. They have a similar action with tumor necrosis factor, but also they have great role in fever. So temperature is elevated by this IL-1. Interleukin 6, they are secreted by macrophage and other cells. They have a systemic effect which causes acute phase response. And what is this? We will be looking at the other slide. Chemokines, they are secreted by macrophage endothelial cells and T cells, mast cells. They recruit leukocyte to site of inflammation. And now you know which chemokines secrete which specific cell, right? Then in chronic inflammation, IL-12, where they are secreted by dendritic cells and macrophages. They cause increased production of interferon gamma. And you know interferon gamma, they are secreted by T lymphocyte and natural killer cell. The function of interferon gamma will be activation of macrophages. Normally, macrophages are not that much active to kill a strong bacteria like tuberculosis. So this interferon gamma make this macrophage into epithelioid cells. Epithelioid cell. 2I between O, epithelioid. Why we call it epithelioid? Because it looks like epithelium. Looks like. It is not actually, when you see under microscope, it is little expanded. It has very vague cell borders, but they are very strong. They can kill mycobacterium. What is the peculiarity of this bacteria, mycobacterium? It has a cell wall which has lipid. So it is very difficult to kill it because it is surrounded by a lipid. So nothing can go in. But by activated macrophage, they can kill this bacteria. So if you have a good immunity and activated macrophage, a possibility that this microorganism might have entered into your body and it has been killed because of your good immune response. Clear? Okay. So they have activation of the macrophage, increase ability to kill microbes, which I said already, and also tumor cells. Sometimes even the newly formed tumor cell, which are just been formed, are killed by these macrophages. Then interleukin 7, which are secreted by T lymphocyte. The function is recruitment of neutrophil and monocyte. So we will discuss this thing while we will be doing chronic inflammation. Yes.
You just mentioned something about macrophage will kill the new one.
Tumor cells. You know, tumor cell, you remember, are I think after inflammatory lectures, Dr. Oban will be taking the lectures for your neoplasia, right? Neoplasia or neoplastic process does not start at once. It has different steps, right? The cell will go first metaplasia, then dysplasia, and different degree of dysplasia, stage 1 or grade 1, grade 2, grade 3, and then it goes into the neoplastic process. And when suppose the cells are in this process and macrophage is activated, it can even kill that cell. So the process can be stopped where it starts. So the person will not have that neoplastic one if you have a very good immunity. Okay, now this is a chart I was talking about. So this TNF1 and interleukin 1, they have local effect as well as systemic effect. So these are the local effects, and this is systemic effect. In local effect, it causes vascular endothelium, expression of leukocyte adhesion molecule. We have already discussed, right? Production of IL-1, which we all know. Increase procoagulant. How? Because when there is damage to the basement membrane, and when basement membrane is exposed, then the procoagulant activity increase, right? Okay, and also decrease the anticoagulant activity. This thrombus topic will be discussed in your next phase, and it will be taught by Dr. Rehan, right? And he will tell you how this coagulation process. We have one slide of coagulation because it is also present in your inflammatory one. I will not go in detail, but Dr. Rehan, while he will be teaching thrombus and embolism, he will discuss everything there, right? In leukocyte, they cause activation and production of cytokines, different cytokines. Fibroblast, this actually acts when we will be discussing the repair process, means next week. Next to next week, yes. So fibroblast, it causes proliferation and also increase in collagen synthesis. We need collagen for the repair process. When the collagen is formed, it is formed from fibroblast. So they excessively secrete collagen to repair that process which has been damaged. And finally, systemic effect, which is acute phase response or acute phase proteins. So it causes fever, it causes leukocytosis, it causes acute phase protein. What are the acute phase proteins? There are many, but three are more important. One is C-reactive protein, number one. Number two, fibrinogen. And number three, amyloid associated protein. So there are three important proteins. The fibrinogen, when they become more high, so if you take a blood of a person and put it for 1 hour in a tube, the RBC comes down, which is called ESR, right? So why ESR is raised in chronic inflammation? Because this fibrinogen, they attach to the RBCs and they form a rouleau formation. What is rouleau formation? Remember, in acute inflammation, I said when there is turbulence, the RBCs come in the center because of rouleau formation, right? So this is in a living thing. I'm talking when you take the blood out, then this fibrin will attach to the RBCs and again they form rouleau formation. And by doing this, the RBC become heavy. So as much fibrinogen will be there, more will be attached to the RBC and there will be more and more ESR level because it will pulling down because the RBC become more and more heavy and it will settle down very much. So the normal value of ESR is 0 to 7 in American books, but here it is almost 12. We considered it even normal. But if in chronic inflammation, the ESR can go up to 50, 60. I have seen one patient having ESR 150. Just imagine. So how much C-reactive, these proteins will be formed, right? Okay. Then we have loss of appetite. You must have seen when you are ill, you don't want anything to eat. So this is a loss of appetite which is caused by tumor necrosis factor and interleukin-1, and also increased sleep. You feel like, don't disturb, please, I'm sleeping. And you know, I have seen patients sleeping for 12 hours without any gap. And when you become all right, you feel yourself strange. Why I was sleeping that much? Because of tumor necrosis factor. And this also causes cachexia in a tumor, right? You must have seen probably any of the family member which had this cancer. They are very thin and lean, right? This thin and lean is because of this tumor necrosis factor, cachexia. Okay. Now coming to a lysosomal constituent, they are present in the neutrophil and monocyte, we know it. And they have lysosomal granules, which is non-oxygen dependent one. This is one picture of a neutrophil. You can see granules. When released, may contribute to the inflammatory response. So we have two primary and secondary granules. The primary, they are also called azurophilic granules, or they are non-specific one. They are myeloperoxidase, lysozyme, acid hydrolase, and neutral proteases. The secondary one, they are called lactoferrin, lysozyme, alkaline phosphatase, and collagenase. Okay, so you must remember these names. These are the lysosomal enzymes. Probably you might be asked question about what are the different lysosomal enzymes. So these harmful proteases are held in check. So they are there, but you had to be check and balance. So for this, there are anti-proteases which are secreted in the body come from the serum and tissue fluid, which is alpha-1 antitrypsin, which is formed in the liver, is a major inhibitor of neutrophil elastase. So whatever destruction is caused by this, so we have anti-proteases, which is alpha-1 antitrypsin, which take care of this lethal effect. Okay, thanks God. So we have both of the things, destroying and also repairing. Now we are here in a complement system. This is a complex slide. So don't be confused with this complex slide. So we have another one, which this in your book, Robin's book. So I will just go back just to make you understand that there are three main pathways to activate this complement system. One is a classical pathway, the other one is alternative pathway, and the third one is lectin pathway. The classical pathway has been activated by antigen antibody, right? The alternate pathway with the microbial cell wall. And the third one is lectin, which it is activated by, again, which is mannose binding lectin. That's why it's called lectin pathway, right? Now, don't go on this, what you call rough light. Just remember that all these processes leads to formation of C3, right? That's why when you ask C1 and the other one, I said, yes, it has a little effect, but we are dealing with C3 and so on. So this C3 is converted into Ca and Cb by the action of which enzyme? Convertase, right? So this C3 convertase changes this C into Ca and Cb. Now Ca goes out and go up and attach to the neutrophil or whatever, and it causes, it attaches with the mast cells and cause degranulation, and it also causes there's a Cb which goes on and then they activate C5. This side C5 again convert into two, A and B. A goes up with C3A, C5A also go there, causes anaphylatoxins, and C5B goes down and they activate, say 6, 7, 8, 9 and form membrane attack complex, MEC. Okay, so this is what written here in your Robin's book. So there are three pathways. They actually form this C molecule, and this is then converted into Ca and Cb. Cb also, if it is not going to attach with C5 to form, convert into C5A or B, the C3b, when it attaches to the microorganism, they act as opsonin. It's strong opsonin, and you know what is opsonization. When it attached to the microorganism, it actually help in phagocytosis. Clear? So this is all function of complement. This membrane attack complex, when it is formed, it actually produces a hole in the cell wall of the microorganism, and if the hole is produced, then what happens? The microorganism get burst out and it dies, right? Okay. So we have clotting factors circulating in the plasma, produced in the liver, coagulation, that is produced, production of fibrin, endothelial cell activation and leukocyte recruitment. So if you just remember this chart, we will not go in detail. Probably Dr. Rehan will be telling you more about thrombosis. We have circulating factor 12 in a body, which is in an inactive form. This factor 12, which is H1 factor, is activated through the collagen, basement membrane, and activated platelets. As I said, when the endothelium is detached, so the basement membrane is exposed. This exposed endothelium, the basement membrane, activate this H1 factor or the collagenase or okay. So this H1 factor is activated by collagen, basement membrane, and activated platelets. So when it is activated, it becomes H1 factor 12A. This 12A goes on the right side and it causes clotting factors, and cascade start by activating this thrombin, and this thrombin which causes fibrinogen into fibrin. Now this fibrin is a clot. So now the clot is formed. So this in one side the clot is formed, and the other side you have to dissolve this clot also. So when there is injury to the endothelial cell, there is a clot formation, and by forming the clot, the body try to regenerate the endothelial cell as soon as possible, right? And as the injury is been done, cleared, what will happen? The clot which is present on the top will be replaced by plasmin, right? So the other anti-clotting system is also going on simultaneously. So what happened here, that one side the clot has been formed, and the other side this H1 factor will act on prekallikrein, and it forms kallikrein. Now this kallikrein goes up and it activates this H1 factor once more, and also it goes down and it causes activation this high molecular weight kininogen into bradykinin. And you know bradykinin is a strong vasodilator, right? This kallikrein also act upon plasminogen, which is inactive form. It forms plasmin, which is active now, and the function of plasmin is to break down this clotting system. So it is, they split this fibrin which is formed. So as soon as the clot is formed on a vessel, and when the vessel is repaired, so the plasmin comes up and it vanishes everything, and everything become normal. So these things are going on in your body and you don't know what is going on, right? So this is a blessing, you know. If you have to control everything by yourself, you will be busy all the time, right? Okay, now what happens then? This kallikrein goes down and it acts on C5 and convert this C5 into C5A. It's also one of the strong vasodilator, right? And anaphylactoxin, right? Now this plasmin has activated fibrin to dissolve clot. It also acts on C3 and forms C3A. Again, this C3A and C5A, they are vasodilator. So this is how this clotting factor is not only doing the clot, but also helping in inflammatory process. Clear? Okay. Now coming to kinin system, you know, bradykinin is produced in the liver, found in circulation as a plasma, potent endothelial dependent vasodilator, and cause contraction of the smooth muscle, increase vascular permeability. Also it involved in the mechanism of aspirin. So you give you aspirin and the pain is decreased. Kinin. Now coming to the general role of all the chemical mediators, last slide. Vasodilators, which are prostaglandin, nitric oxide, and histamine, increase vascular permeability. They are histamine, serotonin, C3A, C5A, by liberating vasoactive amines from the mast cells and other cells. You have bradykinin, leukotriene, C4, D4, and E4, platelet activating factor, and substance P. Then we have chemotaxis, leukocyte recruitment by and activation by tumor necrosis factor, leukotriene, interleukin 1, chemokines, C3A and C5A, leukotriene B4, and bacterial products. Fever, as you know, IL-1 and tumor necrosis factor plus prostaglandin. fever. Pain, prostaglandin and bradykinin. And tissue damage by lysosomal enzyme of leukocyte, reactive oxygen species, and nitrous oxide. So, you must remember with all these mediators, there seems to be an intelligent system of check and balance. As I said, if not, inflammation would take over the world and would run riot. So this is a good saying, and it is written in old books of Robbins, that there is always a check and balance, and in inflammation you need to have check and balance in each and every steps. So this was all about chemical mediators. Any questions from the class, and any question from the online one? Yes.
Yes, so from the online, we have two questions.
Yes.
One is when you're talking about acute phase
Proteins.
Yes, so one is asking, what is acute phase?
Acute phase protein, they are secreted when especially they are secreted in a chronic inflammations, not in acute. Why? Because in acute inflammation, they last for only 48 hours. So they does not have effect on those proteins. But in chronic inflammation, they have effect on these proteins like fibrinogen, C-reactive protein, and amyloid associated proteins, right? And if in a long process, in a chronic inflammation, you may have deposits of amyloid in different parts of the body, right? Exactly, if you see C-reactive protein, they have increase in chronic inflammation. Even in ESR, which I have mentioned the whole story, how this fibrinogen attach with the RBCs and they form a rouleau formation, and the RBCs become heavy. So in ESR, because of that rouleau formation, the ESR will be high. So this is the explanation regarding that. What is the other one?
Thank you, sir. And the other one is you had a picture of the complement system, right?
Yes.
So the students were asking if you can please go over that again.
Okay, I'm going back. Complement. Yeah. So this is a complex slide which is not present in your book, right? Just to show you that how these things are being made. I'll advise you to go through both of the slides, not only this one, which is present in your Robbins book, and a very simple slide, but also go back. So you will be able to understand how this cleavage is going on, how this C3 is being converted by this C3 convertase enzyme to C3A and B, and this C3A, if you see here, C3A after this enzyme C3 convertase, they cleave and form C3A and C3B. C3A goes out, C3B then act on this C5 convertase enzyme, and this C5 will convert into C5A and B. Again, C5A goes out, they act as a anaphylatoxin, and this C5B, they go down and activate C6, 7, 8, 9, and they form a membrane attack complex. Now this C3A and C5A, as I said, they are strong vasodilator, anaphylatoxin. C3B, sometimes if you go in the next slide, this one, C3B, it goes out, and then sometimes then if it goes out, it will not go down. So it go out and they attach with the microorganism and they act as an opsonin. So and the function of this opsonin is to attract this cells, neutrophils or macrophage, to eat this microorganism, bacillus, and they eat it and they kill this bacteria. So this is one of the strong opsonin of this complement system.
So complement from 1 to 4 for opsonization, from 5 to...
No. Only C3B, not all. The all one, if you go back, these 1, 2, 3, 4A, they are actually, one C1 is been converted to C2, C3, 4. Ultimately, we reach to C3. This is an important one, which you had to remember. And then from 3A is forming vasodilation or anaphylatoxin. 3B is a opsonin, right? And if it is not attaching to the bacteria, it will go down and it activate C5. C5 again divide into two, C5A and B. C5A goes up for anaphylatoxin, C5b goes down to activate C6, 7, 8, and 9. 6, 7, 8, 9 are membrane attack complex, right? So this, how it is, how it acts, if you go to the other slide, this 6, 7, 8, 9, they attaches to the microorganism and they form a hole in the cell wall. And this hole, when the hole is formed, what will happen? The whole fluid go into the bacteria and the bacteria burst itself. So it helps in the lysis of the microorganism.
Yes, that's it. So some of the students are asking if you have any tips for memorizing this.
Yeah, I would like to say that, my last slide here, this one. Please kindly look this slide again and again, in which I have written the whole role, what is this chemical mediators are doing. And if you go back in my lecture, if something is deficient here, write by yourself. You make a chart and then just memorize, ask from each other, what are the vasodilators, what are the chemical mediators which are increased permeability, what are chemotaxis one, what are producing fever, what are producing pain. So by this, if you ask from each other, you will memorize. And it is really important. I'll not be going in detail about the molecular concentration or whatever. I'll just ask the functions and this thing. But for your steps exam, the lecture, you had to be very careful to look into it, right? You should know which chemical mediators are doing what thing, right? Is it clear now?
Yes.
So inshallah, tomorrow we'll be starting with the morphological pattern next week and chronic inflammation. Okay, thank you very much.
Thank you.
Can you please take me that?
Yeah, one second.
You have recorded the lecture?
Yes.